And we're going to be talking about genomes, and this is going to be split into two videos. In the first half, we'll talk about how you study genomes, namely through genome sequencing. So, what does that mean? We'll talk about some of the sort of broad things that have been learned from sequencing genomes. Um, and then we'll focus in specifically on what we've learned from studying prokaryotic bacterial genomes. In the second video, we'll focus more on eukaryotic and specifically the human genome, as well as talk about a couple of other techniques, proteomics and metabolomics, which can be used to complement the study of genomes. All right, so we know what a genome is. It's all the DNA in an organism or in a cell. And the way we study a genome is by quote unquote sequencing it, which means determining the order of the nucleotides in all the DNA molecules that make up that genome. So, the order of those nitrogenous bases, A, T, G, C, C, right? That is the genetic code. That's what determines the characteristics of that particular organism. So, key to understanding the genome is knowing its sequence. And we're not going to go into the details of the technology in terms of how that works, um, but we will sort of talk about it broadly in terms of how the technology has changed over time. So, in a nutshell, if you have a genome that's been sequenced, or at least partially sequenced, what can you do with that information? Well, in sort of a broad biological sense, there's some very useful things that could be done. So you can compare species, better understand evolutionary relationships between different organisms, compare individuals even within the same species, identify specific mutations that might be linked to certain phenotypes or diseases. Um, and that is probably arguably one of the most useful practical things that you can do with the genome sequence is study disease and help influence medicine. And we'll come back to that idea when we talk about the human genome. So, as I mentioned, we're mostly going to talk about eukaryotic genomes in the second video, but you can't talk about genome sequencing without first mentioning the Human Genome Project. So, this was a 20-year effort, first put forth in the mid 1980s. The goal of this project was to try to determine the sequence of the full human genome. And the reason I say you can't talk about genome sequencing without this is because this was such a huge, well-funded effort. It really drove the sequencing technology and our ability to sequence genomes more cheaply and more rapidly. Without the Human Genome Project, none of that would have come to fruition. So as I said, this was a 20-year effort. A lot of the money for this came from federal grant funding, although there were also a couple of privately funded research groups that were pursuing the human genome on their own. At the end of this project, some of the publicly and privately funded groups sort of ended up collaborating to push through to the end. But it ended up costing about $3 billion to sequence the essentially 3 billion nucleotides in the human genome, so about $1 a nucleotide in the end. Um, and as our ability to sequence got better and better, it turned out really the first genomes that we were able to fully sequence were prokaryotes, bacteria, because their genomes were much smaller, and then simple eukaryotes, and the more complex multicellular eukaryotes and the human genome really followed after. But I just wanted to point out it was all sort of driven by the Human Genome Project and the intense amount of effort that was put into that. So, how do you sequence a genome? Now, as I said, we're not going to go into all the details of the technology. You can always look up a YouTube video if you're curious in terms of how that works. But in a nutshell, you would have to isolate the DNA from the organism you want to sequence. So purify the genomic DNA. That's pretty easy to do. You then cut that DNA into shorter fragments of about 500 nucleotides, or base pairs is the terminology that's used, in length. And that's done because, especially in the early days of genome sequencing, you just weren't able to sequence in any stretches longer than that. So if you wanted to get complete coverage, you needed shorter fragments. You would then sequence those shorter 500 base pair fragments. And in the case of the human genome, as I mentioned, that's about 3 billion base pairs in length. They ended up with about 6 million of these fragments that had to be sequenced. And then you would essentially use computer software looking for overlaps in the sequences in those fragments to try to piece back together the entire genome. And this effort really led to the development of a whole new subdiscipline of biology called bioinformatics, which is using computers and computer science to try to organize and interpret genome sequences. So you can imagine you're generating a huge amount of data when you sequence a genome. Um, and just being able to organize it, keep track of it, make it accessible to researchers is a big challenge. So that's where bioinformatics comes in. And this is really a hot field in biology right now because so much of biology and biomedical science is pointed towards genomics and our understanding of the genome, that I think there's going to be a lot of jobs in bioinformatics for the foreseeable future. And as I mentioned already, um, our ability to sequence DNA, just the basic technology that's used, advanced very, very rapidly due to this Human Genome Project effort. So we saw this rapid evolution of the technology to the point where now we have what we consider to be high throughput sequencing, meaning we can sequence large amounts of DNA very, very quickly and very cheaply. So just to give you some feel, right, that first human genome project was, as I said, 20 years, about $3 billion. Now you could sequence a full human genome for, let's say, $1,000, maybe even under $1,000 at this point, and in 24 to 48 hours. So that gives you some idea how much the technology has changed over that 20-year period. And I also wanted to point out that as genomes get sequenced, almost all of them are made publicly available on websites. And that's really important because, of course, the value of having a genome sequence is to make it available to researchers that are studying specific genes or specific organisms, and they can use that information to help guide their research. So, what kind of things can you do with a genome sequence once you have it? Well, just in terms of better understanding the genome that you sequenced itself, one thing you can do is you can identify the genes, or what are often called the open reading frames. So this would just be the portions of the genome that actually code for proteins. And we can identify those, or computer programs can identify those pretty easily, because remember, every gene starts with a start codon, ends with a stop codon. It's usually preceded by its own promoter sequence, or other regulatory sequences. So we can find those genes, we can find those regulatory sequences. And then if we know the sequence of the gene, because we have the genetic code, we can estimate the amino acid sequence of the protein that would be coded for by that gene. There are computer programs that can then, from that amino acid sequence, estimate how that polypeptide chain would fold up. What's the shape of the protein going to be? Where in the cell might it be located, and might even give some clues as to the function of that gene, even if you'd never studied it, never seen the protein in real life before. Um, you could also identify some of the DNA sequences that code for things like ribosomal RNA that makes up a significant chunk of the genome. And of course, try to better understand parts of the genome that don't code for proteins, some of those non-coding sequences. There's typically a lot of that in eukaryotic genomes, as we'll talk about. So what is it doing? What's its function? And as we study genomes, it's opened up another subdiscipline of biology or genetics called comparative genomics, which is we can use genome sequences to compare between species or even between individuals within a species. And by looking at the differences in genomes, that can give you some very important clues in terms of differences in phenotype, right? In terms of appearance or morphology or potentially even, behaviors if they're genetically determined. So there's a lot of useful information that could come from just simply comparing genomes across species. And as I alluded to before, this can be very, very helpful in terms of establishing evolutionary relationships between organisms. So, traditionally, evolutionary relationships were studied just by looking at morphology and physical appearance. The idea was the more similar two organisms were, the more closely related that they would be. But of course, we have a lot more detailed information in the genome, but that same basic principle holds true. The more similar two organisms' genomes are, likely the more closely related they are. That's the basis of, paternity type testing and things like that. So as I think I mentioned, the very first genomes that were sequenced, because they were smaller, were prokaryotic genomes. And in fact, the very first genome to be fully sequenced of a free living organism was this bacteria, Haemophilus influenza, in 1995. There were a number of virus genomes that had been sequenced before that because they are even smaller than bacterial genomes. But of course, viruses are not living organisms, so it's a little bit of a different category there. But yeah, 1995, we had the genome of Haemophilus influenza, which is a pathogenic bacteria that can cause pneumonia, often in people that may have influenza, have the flu, right? Hence the name here. And you can see a little graphic representation of the genome here. Remember, bacterial genomes are usually just consist of a single circular chromosome, whereas eukaryotic genomes are made up of multiple linear chromosomes. So here you see the one circular chromosome from H. influenza, and each one of these colored stripes is supposed to represent a different gene or open reading frame. And this particular genome was found to have about 1,700 genes. And what was really amazing was, even though we had been aware of this bacteria for a long time, people had been studying it for a long time because, of course, it causes human disease and there was a lot of interest in it. We found once we sequenced its genome that we only knew what, a little over half of these genes did, what proteins they produced, and what the functions of those proteins might be. So, 42% of this genome, we had no idea what the genes were, what the proteins coded for by those genes did. So, if we really want to understand this bacteria and try to prevent it from causing human disease, all of a sudden we had a lot more work to do, right? What are those other genes doing? Um, so this is a very nice example of what we would call functional genomics, which is using genomes and genome sequences to try to better understand biological function, right? In this case, we want to know what is the function of these 42% of the genes in this genome. What do they do biologically speaking? How do they interact with a host? Another interesting thing we found from studying prokaryotic genomes is the prevalence of what are called transposable elements. And these are sometimes sort of jokingly called jumping genes, but these are DNA sequences that can basically be copied and reinserted in a new place in the genome. So here's your transposable element. We would literally make a copy of that segment and then reinsert the new copy over here. So now we have two copies of that transposable element, and that can be repeated over and over again. And as you can imagine, if you just insert this transposable element DNA sequence in the middle of, say, a gene here, right, that is going to alter and potentially disrupt the function of that gene. So these transposable elements can actually be inducing mutations and causing changes in the genome over time. And so it turns out there's different types of transposable elements. Possibly the most interesting are what are called transposons, sometimes called composite transposons. But these are larger transposable elements that can be about 5,000 nucleotides in length. And they look something like this, where you actually have two transposable elements sandwiching some genomic DNA that might contain a gene or maybe even multiple genes. And this whole chunk, both transposable elements and the genomic DNA in between, can get copied and reinserted. So this, let's say, imagine in a bacterial genome you have a gene for antibiotic resistance here, right? All of a sudden you're creating copies of that gene that may be able to mutate and evolve and give that bacteria ability to be resistant to multiple antibiotics. Another thing we've learned from sequencing prokaryotic genomes is it's made it easier to focus on some of these bacteria that do cause human disease. Because in traditional microbiology, if you wanted to study a bacterium, say one that's causing disease, you'd have to isolate it from the patient. Then you'd have to be able to culture it in the lab, and that may require some very specific culture conditions, specific nutrients, um, specific aerobic requirements. The bottom line is some bacteria are very difficult to grow in the lab or impossible under current techniques to grow in the lab. Whereas with genomics and genome sequencing, we can use some of these techniques to basically just isolate DNA from the bacteria, sequence it, identify the bacteria just using its DNA without the requirement for culturing. And this has actually led to another subdiscipline of biology called metagenomics, which is just studying environmental diversity, mostly microbial, things we typically can't see, but environmental biological diversity just by collecting environmental samples, isolating DNA, sequencing that DNA, and then comparing it to these large databases that we have of sequenced genomes. So there's thousands of genomes that have been sequenced at this point, and using that to identify either existing samples or existing species in the sample, or potentially even new species. And so as we've started down this journey into metagenomics, it's been estimated that about 90% of the microbial world has been unknown, right? It's just things we couldn't see them, we didn't know they were there, they haven't been able to be cultured in the lab, but now through genome sequencing we can quote unquote see these bacteria and start to identify them. Another very cool idea that's come out of prokaryotic genome sequencing is the concept of universal genes and a minimal genome. So, as we've sequenced more and more bacterial genomes, we can compare those genomes and say, what are the genes that we see in basically every living cell? We would maybe consider those to be, required for life. Whereas if there's other genes that maybe just pop up here and there, those might be sort of more specialized genes imparting some very specialized function. So, what researchers have been able to do is identify these universal genes, and basically whittle the bacterial genome down to this minimal set of DNA sequences, regulatory sequences, and genes that have now been shown to be capable of keeping a cell alive, at least under certain specific laboratory conditions, and allow that bacteria to propagate and divide. And what's really cool about that is it facilitates a lot of genetic engineering, so of microbes. So if you have this bacteria that has this minimal genome, you can then kind of go in and customize the genome by adding other specific genes to give that bacteria features you want. And there's been some ethical debate about that, because of course you could do great things with that, right? We could sort of make designer microbes that would help, clean up oil out of water or something like that. But you could also just as easily create designer microbes that would be very, very pathogenic and potentially very harmful to people. So I'm going to stop there and we'll pick this up in part 2.